Hello students, good morning. I'm your KVR sir. Today we came with a new topic, that is biotechnology in one shot. Biotechnology is one of the important lesson. None. Biotechnology is nothing but gene manipulation. We will change kind of alterations, whatever kind of alterations are required, we can make in a gene to make our product in our wish. Okay, so biotechnology, genetic engineering is mainly connected with gene manipulation. Clear? And so basic fields where the biotechnology now working is in vitro fertilization, leading to a test tube baby formation, and synthesizing a gene, artificial synthesis of gene. You know very well, we use PCR technique, polymerase chain reaction technique, for the synthesis of gene, and developing DNA vaccine and correcting a defective gene. So in we have discussed that 4 years old girl, severe acute SCID, some disease, so she have transferred one gene. So correcting a defective gene is also one of the upcoming technology. So defective gene can be altered or given by a new gene itself. Okay, these are the 4 major parts we are working on according to our textbook scope. And what is the definition of biotechnology given by the European Federation of Biotechnology? The European Federation of Biotechnology has given the definition of biotechnology. It emphasizes both the traditional view and modern molecular biotechnology. The definition of biotechnology is given as integration of natural sciences with organisms. So one who reads this statement as a whole will feel it is a bit difficult. So bifurcate it. So the first word is integration of natural science with organisms, cells and their parts thereof, and molecular analogs. So this is organism, and lesser than the organism we have the cell, lesser than the cell parts thereof, part of the cell, that is nothing but chloroplast or mitochondria or nucleus, or molecular analogs, that is nothing but DNA and RNA. Are you getting me or not? Integration of natural science with organisms, cells, parts thereof, and molecular analogs like DNA and RNA, and for the products and services. Was it clear? Integration of natural science with the organisms, cells, parts thereof, and molecular analogs for products and services. Clear? So this is the definition of biotechnology. And coming back here, the biotechnology or genetic engineering have 2 fundamental principles. The one is genetic engineering. What do you call it as? Upstream processing. Genetic engineering is nothing but upstream processing. Whatever you do to make a recombinant bacteria, a recombinant cell, so up to that is genetic engineering. So concerned with the scientific knowledge is essentially required for that. After making one recombinant bacteria or a recombinant cell, a recombinant host cell, and maintain the sterile ambience and provide optimal conditions for that recombinant bacteria to produce our products, that is called as downstream processing or bioprocess engineering. Was it clear? First upstream processing, next downstream processing. First, in the upstream processing, we prepare a recombinant bacteria. Okay, we can call it as recombinant. Even recombinant DNA preparation comes under upstream processing. First, you will prepare recombinant DNA. Recombinant DNA means plasmid. This is the plasmid. if you introduce your gene of interest in the middle of the plasmid, now the plasmid combination is changed. Now we call it as recombinant DNA. Recombinant DNA. Was it clear? Could you able to see this gene is inserted in the middle of the plasmid? Plasmid plus with the desired DNA is rDNA. Preparation of rDNA or preparation of recombinant bacteria is the first step. First step that is called as upstream processing or genetic engineering. Upstream processing or genetic engineering. The second step, so you have prepared rDNA and introduced it into a bacterial cell. Now you call it as recombinant bacteria. For this recombinant bacteria, instead I will say this is an insulin gene you have inserted into a plasmid, and then that plasmid is introduced into a bacteria. Now the bacteria is said to be recombinant bacteria. This is insulin gene isolated from pancreas, Langerhans islets of pancreas. That insulin gene is introduced into the plasmid. Now we call it as rDNA. What do you call it as? rDNA. rDNA is now introduced into the host organism or bacterial cell. Now the bacterial cell is said to be the recombinant bacteria. Recombinant bacteria. So up to the production of recombinant bacteria, it comes under genetic engineering. Genetic engineering. So now this bacteria requires the fermenters. Big fermenters are required. So that a bacteria can replicate itself and it can synthesize. This is fermenter. So temperature, foam control, pH control, sample port, everything is there. This is bioreactor. Bioreactor, you have to provide sterile conditions for the bacteria to grow as well as to synthesize insulin. Was it clear? This production of insulin is called as downstream processing or bioprocess engineering. So there are two fundamental principles are there. The one is upstream processing or genetic engineering. That scientific knowledge is essentially required. So bioprocess engineering, if you give a recombinant bacteria to a bioreactor or a fermenter, so the product is produced. So isolation, purification and preservation of the product, everything comes under bioprocess engineering, that is downstream processing. Clear? Then let's go with the tools and techniques of the biotechnology. What is essentially required? What is essentially required for biotechnology? So for our rDNA technology purpose, the first is restriction endonucleases and polymerases, DNA polymerase, RNA polymerase, and DNA ligase, vectors, and the host organisms. These are the tools required for biotechnology process. Done. So let you go with the first word, restriction enzymes. In the year 1963, the two different enzymes were isolated. Two enzymes, 1963, two enzymes responsible for restricting the growth of bacteriophages in Escherichia coli were isolated. So 1963. One of these is methyl groups to DNA, addition of methyl group to the DNA. So see here, this is the bacterial cell. And so this is one virus ready to attack the bacteria. Bacterial attacking viruses are called as bacteriophages. Clear? This is bacteriophage. So this bacterial DNA synthesize lots and variety of different types of restriction endonucleases and exonucleases as a defense enzymes. These enzymes will not allow the viral DNA. If virus is attached here, so first you know the very well different steps, viral replication, different steps is there. First step is adhesion of viral cell to the bacterial cell. Second step is penetration. The DNA will enter into the bacterial cell. Third step is this viral DNA will disintegrate the bacterial DNA so that you cannot see either virus or bacterial DNA. A bacterial cell with viral DNA is a condition called as eclipse stage, and finally new virion particles are synthesized and comes out by breaking down the bacterial cell is called as lysis. Okay, there are the steps not required right now. As soon as the viral DNA enters, these enzymes will cut the DNA from one end to another end. That kind of enzymes are called as exonucleases. Or else the viral DNA is cut in the middle. That is called as endonucleases. So these kind of enzymes will restrict the growth of viral DNA in the bacterial cells, hence called as restriction endonucleases. Are you getting me? What are the restriction endonucleases and exonucleases? These are nothing but defense enzymes. Defense enzymes that protect bacteria from viral DNA. So different types of defensive enzymes will be synthesized by the bacteria to protect itself from the viral DNA. As soon as the viral DNA comes, it will disintegrate the bacterial DNA. Before that, bacterial DNA has synthesized many different types of enzymes that may cuts the DNA from one end to other end. This is called as exonucleases. Will break the DNA into nucleotides. Okay, sometimes it may cut the viral DNA in the middle. Endonucleases. Both exonucleases or endonucleases together called as defense enzymes. So we use this restriction endonucleases to cut our DNAs wherever DNA is supposed to be cut. So this is a long DNA isolated from one chromosome. I want this piece only. So for this cutting of these enzymes, I will use restriction endonucleases. And what he is discussing about this methyl groups and DNA. So these enzymes, if they are cutting the viral DNA, they can have the same property on the bacterial DNA also. So how could the bacterial DNA get protected by the self enzyme like lysis? So these enzymes, if cut the viral DNA, how the bacterial DNA get protected? By terms of methylation at certain places. So 1963, the scientists isolated two types of enzymes. One is methyl groups added to the DNA. What do you call it as methylases? Methylase. That will addition of methyl group to the bacterial DNA so as to protect from the defense enzymes, self-digestion. Okay. And the second, the latter was restriction endonucleases. Do you now understand what do you mean by restriction? So the enzymes that will restrict the growth of fast DNA into the bacterial cell, either by cutting from one end to another end, that is called as exonuclease, or cutting by in the middle, that is called as endonucleases. So this discovery of restriction endonucleases is a revolutionary step in the biotechnology process, because unless we have that restriction endonucleases, we cannot cut our desired DNA from the particular source. Restriction enzymes helps us to cut the DNA, to isolate a separate gene or a fragment of the DNA or a separate desired gene from the large source of DNA. Hence, restriction endonucleases are called as molecular scissors or chemical scalpels or DNA scalpels or molecular scissors, chemical cutters. Okay, the first restriction endonuclease was discovered as HIND2. Okay, clear? Let's go to the next slide. The function depends on a specific DNA nucleotide sequence and isolated and characterized 5 years later. Okay, so what exactly the function of the restriction endonucleosides? HIND2 is always cuts the DNA molecule. HIND2, remember, this is the first enzyme discovered So by recognizing a specific sequence of 6 base pairs. Okay, and this specific sequence is known as recognition sequence for the Hind2. So these restriction endonucleases, for example, let us say this is the enzyme restriction endonucleases. They cannot cut the DNA like wherever they want. Sorry to say, they cannot cut the DNA every place randomly. They will cut the DNA at a specific site. They will have particular sequence of 6 bases will be there. That is called as recognition site or restriction site. Was it clear? They can cut the DNA on both strands at a specific site only. A site of 6 base pairs, 1, 2, 3, 4, 5, 6 base pairs. This particular sequence is called as a restriction site. Clear? So restriction endonucleases cuts the DNA at a specific sequence. What do you call it as? Restriction site or recognition site. Today we have more than 900 restriction endonucleases isolated from 230 different strains of bacteria. So do remember, restriction endonucleases are present in some prokaryotic bacteria and yeast also. In yeast also certain restriction endonucleases were produced, because yeast will also be affected by the virus. What do you call it as yeast gymophages. They will attack yeast. Okay, so restriction endonucleases, not only they are isolated from prokaryotic, they are also isolated from eukaryotic cell, that is yeast. Leave it aside. So 230 variety strains of bacteria. So for 900 restriction endonucleases were isolated. And each, what is the function of the recognition site? Each recognize different recognition sequences. So if some restriction endonucleases are there, one I will say ECO R1, HIND3, or BAM H1, Bacillus amyloliquefaciens. So H is the strain and one is the serial number. So different restriction enzyme, Haemophilus influenzae and Bacillus amyloliquefaciens, Escherichia coli. Different bacteria may produce different restriction endonucleases. Okay, these have specific sites. These sites have specific recognition sequence. The conversion of naming of these enzymes, the first letter is comes the genus. E stands for Escherichia, CO stands for coli species, R stands for strain, and 1 stands for serial number, that is isolated from the bacteria. So first letter name comes with the genus, E means ECO R1. Second 2 letters comes with the species, that is coli, and the prokaryotic cell from which they are isolated. For example, ECO R1 comes from Escherichia coli strain RY13, and in ECO R1 the letter R is derived from the name of the strain. Clear? Next, Roman numbers following by the number indicates the order which there. So ECO R1, ECO R2, ECO R3 means that is ECO R1. So ECO R2, ECO R3 means this is the serial number that is isolated from the same bacteria, from its isolated bacteria. Restriction enzymes belongs to a large class of enzymes called as nucleases. Nucleases, because they break the nucleic acids. Hence they are called as nucleases. So these are 2 kinds of nucleases. As I told earlier, one is exonuclease, that cuts the DNA from one end to another end. Another one is endonuclease. For example, if you see this is the DNA and this is another fragment of DNA. If you use exonuclease, it will cut all phosphodiester bonds from one end to other end. So simple nucleotides will be available. But endonucleases will have specific recognition sites. They will cut the DNA in the middle of the DNA, DNA. So because if we cannot use exonucleases for isolation of our desired DNA, because desired DNA will be broke down into nucleotides. We cannot use exonucleases. Okay, endonucleases we can use. Exonucleases remove nucleotides from ends of the DNA, whereas endonuclease makes cuts at specific positions within the DNA. Each instruction endonuclease functions by inspecting the length of the DNA sequence. For example, one enzyme is there. Here is one enzyme is there. So that enzyme slowly binds the DNA and goes slowly, for example, slowly inspect the DNA. For example, if it is EcoR1, you have GAATTC, GAATTC. This is the specific recognition site. So this enzyme slowly sequence the DNA, and then whenever the restriction endonuclease site comes, there only it will bind and break the DNA in between G and A. In between G and A. Was it clear? In between G and A. So one, it finds specific recognition sites. It binds the DNA and cut the DNA on two strands, and both the strands of the double helix. Cut here in the upper strand as well as lower strand. So at specific point, it breaks the sugar phosphate backbone. It breaks restriction endonucleases breaks what? Sugar phosphate backbone. Restriction endonucleases will break the sugar phosphate backbone. Was it clear? Gaurav Insan. Hi Gaurav Insan, coming to you. Wait for couple of minutes. आप वीडियो देख रहे हैं ना? You are watching the video and take it down as it is in your notebook. Shall we go ahead? Gaurav Insan. Next, restriction endonucleases recognize a specific palindromic sequence. Palindromic sequence is MADAM. It gives same meaning in both sides. Madam. Okay, it's a palindromic word. A palindromic word will have similar letters in this forward direction as well as backward direction. Restriction endonucleases, generally they are palindromes. It's a true reading in 3' to 5' direction and 5' to 3' direction. They will bind here, they will cut the both DNA strands, leaving sticky ends. Because the sticky ends are essentially required for the ligation property. Gaurav Insan, आप वीडियो देख रहे? Priyanka Bantu. Priyanka, you are from which state? So are you watching this video? Please write it down in your notebooks. Okay, all points are from our NCERT textbook only. Sticky ends are helps in ligation by DNA ligase. Clear? Done. Next we go. But between the same bases opposite strands, it leaves single strand called as sticky ends. There are overhanging stretches called as sticky ends at each strand. For example, the sticky ends are essentially required. If you cut a gene from the same restriction endonuclease and cut the plasmid with the same restriction endonuclease, you cut the plasmid with the same, for example, I say EcoR1. If you are used to isolate the gene with sticky ends. Okay. So you have to cut the plasmid also with the same EcoR1 so that the sticky ends will be similar, so that the gene can be ligated easily by DNA ligase. Was it clear? This plasmid DNA and desired gene should be cut by the same restriction endonuclease so as to have the similar DNA sequences. Clear? Was it visible to you? So then let us we go for the next enzyme. The sticky and stickiness of the ends facilitate the action of the enzyme DNA ligase, which will ligate our gene of interest along with the plasmid. Now what happened? Recombinant DNA is formed. Recombinant DNA is formed. Was it clear? Done. Let us we go to the next slide. So as we are using the restriction endonucleases to cut the DNA, we call it as molecular scissors. Molecular scissors or chemical scalpels or molecular knives. Molecular knives, DNA cutters. Different names are there. Clear? Some enzymes are used to fuse the DNA fragments. This is sticky end, this is DNA ligase and helps in the ligation. So this is opposite enzyme. The one enzyme is cutting, that is restriction endonuclease. One enzyme helps in attaching to DNA fragments, that is DNA ligase. This is restriction endonuclease, this is DNA ligase. So endonuclease cuts the DNA, DNA ligase allow the ligation of DNA fragments. Clear? Next, separation and isolation of DNA fragments. The cutting of DNA by restriction endonuclease results in the fragments of DNA. First, you have to isolate the DNA in a test tube. Clear? Okay. So in a test tube, large quantity of DNA will come. Large quantity of DNA will come from a eukaryotic cell. So first of all, you have to add restriction endonuclease. Restriction endonuclease. So DNA is fragmented. DNA is fragmented. Do these fragments will have the same length or different length? The fragments will have different length. Different length. Was it clear? So the fragments of different lengths are possible because the restriction endonucleases sites are not evenly distributed. They are randomly distributed, so that a fragment, one fragment is very large, one fragment is very small. So what do you call it as? A restriction fragment. A restriction fragment length polymorphism. What do you mean by? The restriction endonuclease, while acting on the large quantity of DNA, it will produce the fragments of different size. Different sized fragments show polymorphism. Polymorphism means all are not of same size, different size. Different sized fragments are called as restriction fragment length polymorphism. Since the DNA fragments are negatively charged, now we will have to separate. We have to separate the DNA fragments according to our requirement. Was it okay? By using gel electrophoresis. Gel electrophoresis. The technique, agar is used. Agar is extracted from two red algae. This is Gracilaria and this is Gelidium. Red algae, Gracilaria and Gelidium. Two red algae are there. Agar gel technique. DNA fragments are negatively charged. They can be separated by forcing them to move towards to the anode, positive charge, under electric field through a medium or a matrix. So nowadays most commonly used matrix is agarose. So which is a natural polymer extracted from seaweeds. What are the two seaweeds here? This is Gracilaria and Gelidium. These two species are nothing but the red algae. Red algae. Was it clear? Now let us see. First we have isolated the DNA and we have fragmented the DNA. Now let us see. So this is isolated. Large quantity of DNA is obtained by using any forceps and needles. If you isolate the DNA, what is this called as spooling technique? What do you call it as? Spooling technique. Okay. So we will use agar gel. So see this technique. We will make wells here. We will load the DNA. We will load towards to the cathode side. The DNA fragments will move towards to the anode side. So have a look here. So this is agar gel. So make wells here by using a comb. Wells. This matlab patla. This is delicate jelly chocolate like agar gel. Okay, so we will prepare and we will load by DNA by using micropipettes. Okay, so 3A, B, C are 3 wells and D is sample well. Okay, I know the insulin molecular weight is some X. So I will put the same molecular weight DNA in the sample port, sample well, and this DNA will go up to here. So now I have loaded the pancreas DNA, which is isolated from Langerhans islets of pancreas, into 3 different wells. The DNA fragments, smaller fragments will move faster and larger fragments will have lower mobility. Okay, small fragments. This is positive anode side. This is negative. Negatively charged DNA loaded towards to the negative charge. Okay, moving to the towards to the anode based on their size. Based on their size, larger fragments will have slow mobility. Smaller fragments will have faster mobility. Okay, from this well, can you now see from sample port well C, the same sized DNA is obtained. So you have taken sample well, an equal molecular weight DNA is put in the sample well, and you have in this well you have dumped all the DNA fragments. So smaller fragments will move fast, larger fragments will move slow, and a fragment of equal known molecular weight you have loaded in the sample, it will reach here. An insulin gene also expected to reach there. You will isolate the DNA from the agarose gel. This is called as elution. Isolation of DNA from agar gel is called as elution. Clear? So now that isolated DNA is attached with the plasmid. This is called as rDNA preparation. This is called as cloning vectors. So now we are taking the cloning vectors, either bacteriophages or plasmid DNA can be used as cloning vectors or gene cloning vehicles. They will carry, this is bacteriophages, they will carry our gene of interest from one organism to another organism. Hence they are called as cloning vectors or gene cloning vehicles. Okay, and that gene cloning vector or a plasmid DNA should have some ideal features. That is nothing but origin of replication, the point where origin replication starts. Next, selectable markers, it should have selectable markers so as to select the non-transformants and transformants from the colony itself. Okay, and cloning sites should be there. One plasmid should have multiple cloning sites, MCQ. It means the one plasmid vector should have a sequence for EcoR1, BAMH1, HEND3, HED3, small one. So like many sites should be there. When and where I require, I can cut the plasmid DNA at anywhere itself by having the specific restriction endonuclease site. This is called as, I can add my gene of interest in the EcoR1 site, in the BAMH1 site and SAL1 site, or any site itself. So this plasmid should have different restriction endonuclease sites one time only. One EcoR1 should be present at one time only, so that it can cut like this. Our gene of interest will go and attach. Now it is called as rDNA. This is so multiple cloning sites, MCQ should be there. So this is all about some tools and techniques of recombinant DNA technology. In brief, in the next class, we'll learn some more details. Thank you very much for today. Thank you. Have a good day.